Hi, my name is Josiah Baumer, and this is Jackson Summers, and we did a comparative analysis of microbacteriophages Jahseh and Makinzy.
Bacteriophages are the most numerous and diverse biological entities on Earth. And with this in mind, the purpose of our project was to investigate this diversity of the bacteriophage population through isolation, characterization, and comparison between new bacteriophages isolated from the environment. Through the use of our host bacterium, Microbacterium florarium, bacteriophages Jahseh and Makinzy were enriched from different regions of Kentucky. The virus particles were observed using transmission electron microscopy, and both phages have very similar morphological features as displayed in a later slide. The genomic DNA of the phages then isolated and analyzed through a restriction digest of the DNAs and gel electrophoresis. And this analysis has demonstrated that these phages are unique. However, they were grown on the same host and were still isolated from very similar environments. The sequence of Jahseh was recently completed and compared to all other phages in GeneBank, which is the National DNA Sequence Database. And this analysis displayed that Jahseh belongs to the EE cluster of microbacteriophages, a cluster with only 53 members. This project overall has contributed useful information to the scientific community, but additional sampling and analysis will be necessary for us to all be able to develop a deeper understanding of bacteriophage diversity.
There was a deep system of methods that we used in order to get all the results that we did. First, we had to use isolation to isolate the phages that use Microbacterium florarium through an enriched soil sample, which we collected throughout the state of Kentucky. Then we had to purify the phage to get only one particular phage. Then we amplified the number of phage to increase the titer in the phage lysate through creating and collecting webbed plate lysates. Then we viewed the phage through electron transmission microscopy. And then we extracted the phage DNA in order to begin the characterization of the genomes. And then we characterized them using the DNA restriction digest and gel electrophoresis, which separates the phage DNA based on size using an electric current.
So as seen here in both images, these are the plaque morphologies of both phage Jahseh and phage Makinzy. And in a later slide, we have a table filled with the dimensions and respective sizes of these phages.
Here we can see the results of our transmission electron microscopy. And as you can see, the morphological characteristics of both Jahseh and Makinzy had very similar characteristics with that round head and the curly tail as well.
And in these two images, the gel electrophoresis results are easily able to be seen. And as displayed in both, in lane 6, the restriction enzyme HAE3 cut much more significantly than the other lanes.
As you can see here, we did a pretty in-depth analysis of the comparison between the two phages. And so phage Jahseh was collected in Paducah, Kentucky through a soil sample, and phage Makinzy was collected in Bowling Green, Kentucky. And while phage Jahseh had a 2 millimeter plaque diameter, phage Makinzy had a 3.5 millimeter diameter, and both phages were found to be lytic. However, phage Jahseh had bullseye plaques, while phage Makinzy had clear plaques. And then here you can also see the actual dimensions of the phages. And you can see that on average, Makinzy was a little bit larger. Um, Additionally, phage Jahseh and Makinzy both had A3 cut once when in restriction cut sites.
As displayed here through this image, Josiah, Baumer, and I both used Wolfram Mathematica to visualize the locations and coordinates of each phage and its isolation location. Phage Makinzy can be seen in purple, and Jahseh can be seen in the blue. However, the rest of the orange markers are of the EE subcluster of phages, which is the family that Jahseh is in.
In conclusion, we found that Jahseh and Makinzy have distinct morphological characteristics, and then after doing the DNA analysis, we can see some similarities and differences in their genomes. Additionally, our analysis of Jahseh and Makinzy shows that the phage population is very diverse, given all of the differences and similarities between the two.
So our work was conducted through the Howard Hughes Medical Institute's C-Phages program through WKU. And we would like to personally thank Dr. Rodney King for his special assistance in the lab and through providing all the materials. And an additional thank you to Dr. John Andersland for his assistance and teachings with the transmission electron microscopy.